you can memorize things. Okay? I want to just kind of look at them and give some examples about them, and we'll see some specific ones throughout the term. So I'm going to put a table up here in a minute that shows it, but there are probably, by now, probably upwards of 100 or so identified neurotransmitters. When I was taking general biology in the 1980s, I think we had identified, well, science had identified maybe 15 or 20. So neurochemistry, right, which is what we're talking about here, right? Neurochemistry, especially central nervous system neurochemistry, neurotransmitters in the brain, huge. It's a very big, rapidly developing field, and we're learning probably that and sort of molecular biology in general is the area of biology that we're learning so much about so rapidly. So this idea of neurochemistry is really trying to identify and understand the effects of neurotransmitters. That's what neurochemistry really is, right? Trying to do that. They've identified them so far, and they're categorized them based upon their chemistry. And the reason we categorize them based upon their chemistry is that dictates how they work, right? Where they can, do you have to have a cell surface receptor, can it be inside the cell or whatever, right? So it's their chemistry that dictates how they get to their targets. The first neurotransmitter that was identified, and the one we know the most about, is acetylcholine. And the reason we know the most about it is because it is the neurotransmitter that is the only neurotransmitter that is released by somatic motor neurons onto skeletal muscle. So what I just said was all of my somatic motor output releases acetylcholine onto skeletal muscles, and skeletal muscles contract in response to that. So it's very well understood in that regard. It's also, so it's always excitatory in that, under that setting. But it's also been identified as a central nervous system neurotransmitter, meaning if I say something is a CNS neurotransmitter, what that should tell you is that an interneuron or an association neuron releases it, and an association neuron or an interneuron has a receptor for it. So it's totally within the brain or the spinal cord. And in that, acetylcholine is also a CNS neurotransmitter, and it can be excitatory, it can be inhibitory. What I just said by that is it can cause depolarization, or it can cause hyperpolarization. It can trigger action potentials, or it can inhibit action potentials. It depends on the receptor. And you have to realize this, right? The response you get in the postsynaptic cell doesn't depend on what neurotransmitter is out there. It depends on what receptor is recognizing that neurotransmitter. That's what's going to trigger the response. They can be amines. Amines are basic in terms of their chemistry, and they have amino groups or amine groups as their predominant functional group. And we'll look at some examples of amines like dopamine. They tend to end in amine, things like that. You may have heard of those. Amino acids, simple amino acids, can be neurotransmitters. Glycine, for example, can be a neurotransmitter. There's this amino acid called GABA, which is not an amino acid we use in proteins, but it is an amino acid, GABA. And that is the CNS's most common inhibitory neurotransmitter. GABA is. GABA and glycine both. Okay. They can be peptides. Peptides are strings of amino acids. These can be as complex as a protein, so a large, 100 or more amino acids long with secondary and tertiary structure to be it. Or it could be 2 amino acids hooked together, 4 amino acids hooked together. It depends. Right? But it's going to have that peptide chemistry. Purines are all derived usually from ATP. ATP, you know ATP, right? Adenosine triphosphate. That is a chemical class of a nucleotide called a purine. In DNA, right, you have A, G, C's, and T's. A's and G's, adenine and guanine, those are purines. They're a larger 2-ring structure. Sometimes we modify those. In fact, if you modify ATP, you can turn it into a particular neurotransmitter. And that makes sense, right? We have ATP, why not use it as a neurotransmitter? Some of them can be lipids and some of them can be gases. What's unique about these two is that they're nonpolar. And it means that they can diffuse into their target cells. So what's interesting about these two is that do they need a receptor out here? No. In fact, they're going to come inside the cell and trigger a response. their chemistry is actually really interesting in the way they work. You can't store them very well because they are hydrophobic. They would just move through cell membrane. So they tend to be synthesized and released on demand. Gases, the most common one that we use is nitric oxide, and it is a vasodilator. So we use it to trigger smooth muscle relaxation. So as such, it's an inhibitory neurotransmitter, right? It inhibits smooth muscle contraction, blood vessels dilate, increased blood flow, that kind of thing. This is the one that Viagra triggers a release of nitric oxide. We talked about Viagra before with you guys? Maybe not. Maybe it was a different class. But anyhow, Viagra